Hello, my name is Lisbeth, and today we'll be covering SLL 5, which is explaining the structure and the function of the proteasome in normal physiology and disease, as well as recognizing disorder associated with chromosomal dysfunction. First, we're going to talk about how cells break down proteins. So basically, in order to maintain homeostasis, cells must continuously recycle proteins. Proteins can become damaged, misfolded, or simply no longer needed, and their accumulation will disrupt normal cellular function. To prevent this, cells rely on two major degradation pathways. We have lysosome, which degrades around 20 to 10% of proteins. They're mainly extracellular or membrane components. And then we also have proteasomes, which are responsible for around the 80 to 90% of intracellular protein turnover. Like we remember in class, we'll be focusing on the 20S proteasome, which is a highly regulated, energy-dependent complex that selectively degrades unwanted proteins, allowing the cells to maintain protein quality control as well as adapt to changing conditions. Now we're going to talk about proteasome structure, but more specifically the 20S proteasome, which we can see in the image below. So the 26S proteasome is made up of two main components. We have the 20S core and two 19S regulatory cap, which one can be found on each end. The 20S core is a barrel-shaped structure that contains the A and B subunits. This is where the actual activity occurs, meaning that this is the part that physically breaks down proteins into smaller peptides. On both ends, we have the 19S regulatory caps. These caps have three critical roles. They recognize proteins that have been tagged with ubiquitin, signaling that they need to be degraded. They also use ATP to unfold proteins, since the proteasome can only degrade unfolded polypeptide chains, and they regulate access to the core, ensuring that only the right proteins are degraded at the right time. Think of the 19S caps almost like security checkpoints where they have to be properly tagged and unfolded proteins are allowed to be broken down by the 20S core. Okay, now we're going to focus on protein tagging. So before proteins can be degraded by the proteasome, they need to be tagged with a small protein called ubiquitin. This tag acts like a molecular label telling the cell this protein's time is up and it's ready to be degraded. The process involves three main enzymes working in sequence. We have E1, which is ubiquitin activating enzyme. It uses ATP to activate the ubiquitin and prepare it for transfer. We have E2, which is ubiquitin conjugating enzyme, and it carries the active ubiquitin and delivers it to the next step. And then we have E3, which is ubiquitin ligase. This is the key in decision maker. It recognizes the target protein and attaches the ubiquitin to it, determining which protein gets degraded. So you can think of E1 as the energy source, and then we can think of E2 as like the carrier, and then we have E3, which is the quality control, who decides exactly which proteins are meant to be marked for destruction. Now we're going to talk about the functions of the proteasome. So the proteasome plays a central role in maintaining cellular health and function. It is far more than just some type of trash disposal system within our bodies. First, in the immune function, the proteasome degrades viral protein into small peptides. These peptides are then presented on the cell surface by different molecules, allowing the immune system to recognize and eliminate infective cells. Second, in the cell cycle control, the proteasome ensures that regulatory proteins are degraded at the right time. This precise timing helps cells process smoothly through each phase of the cell cycle. And finally, in protein control, the proteasome eliminates any type of misfolds or damaged protein, especially those accumulating in the endoplasmic reticulum. This prevents cellular stress and maintains homeostasis. So the proteasome is not just something that cleans up, it's an actively regulating critical process that keeps the cell and ultimately the organism healthy. Now we're going to talk about some different disorders that are caused by proteasome dysfunction. So when the proteasome fails to function properly, proteins that should be degraded start to accumulate inside the cell. This buildup can have serious consequences for human health. One group of conditions is the proteasome-associated autoinflammatory syndrome, which is PRAS, and some of the ones that you can find under this is things like things like Nakajo, Nishiruma, and Candle syndromes. These are rare autoimmune recessive disorder characterized by chronic inflammation, skin rashes, fever, and even fat loss due to immune system's overactivation. On the other hand, there's disorders like neurodegenerative diseases like Alzheimer's, which deals with defective proteasome activities that allows toxic proteins to accumulate in neurons. Over time, this leads to cell damage and loss of brain function. So a defective proteasome doesn't just disrupt protein cycling, it directly contributes to inflammation, aging, and neurodegradation. Okay, now we're going to go into the NBME style question. So, Bailey researcher is creating a mutant cell which is lacking the E3 ubiquitin activity. Which of the following processes will most directly be impacted? A. ATP hydrolysis for protein unfolding. B. Endocytosis of membrane receptors. C. Target degradation of misfolded proteins. Or D. Transfer of active ubiquitin. I'll give you 5 seconds. If you chose C, you are correct. Because remember, E3 is the one that selects for specific protein. And without E3, proteasomes can degrade specific misfolded protein. Okay, that'll be all. Thank you for listening and have a great day.